So an amino acid, depending on what kind of scientist you're talking to, can mean different things. When I hear amino acid, I picture a carboxylic acid with an amine group on the molecule. That to me could be any number of things, right? I can just draw whatever I can want to, as long as it has those things. You can designate along a chain, say you're on a chain, or even in a ring this would work, the position of the amine group relative to the carboxylic acid using the common designators that we're used to, which is alpha, beta, gamma, delta. Obviously, if you have 10 carbons out, that alphabet system falls apart. So we're talking about the amines that are fairly close to the carboxylic acid. This brings us to, when I mention an amino acid to, say, a biologist, they are not going to think of the same thing I think of. I think of this. If you told me, draw an amino acid, I would say, okay. There you go. That, to me, is an amino acid. They would never come up with that because what they're used to working with are proteins found in animals, right, which contain 20 amino acids, and all of them are, whoa, come back. Let's undo that. All of them are alpha amino acids, and I'm going to abbreviate that AA. So biologically, the alpha amino acids are the ones that are important, not necessarily what an organic chemist would throw out of their head. So an example of an alpha amino acid is this one right here. You can see in it the carboxylic acid unit, which is there. This is the alpha carbon, and then there's an NH2 off of it. So that's an alpha amino acid. And what we're going to see in just a minute is almost all of them have also on that alpha carbon a branch, which is some R group. And so what we're going to be doing for the rest of the amino acid chapter, we're going to be focusing on the alpha amino acids and not on what my version would be. We've already sort of talked about this enough. You know, we're good with amines, we're good with carboxylic acids, we're done. Okay. Now as far as that alkyl group, the side chain group R, it could be any number of things. It could be neutral, like this one's an isopropyl group, nothing very special about it. So that R group could be neutral. We're going to see several examples where it can have an acid on it. Hang on one second, Adam, I see you. It could be acidic, so it will have a second carboxylic acid on it, if that's the case. Or it could be basic. And typically, if it's basic, what it has off of it is another amine whose lone pair is available to go grab a hydrogen. Okay, so we'll see all those examples. Yes, Adam?
The amino group and the amino acid always primary? Or,
the amino group is always going to be an NH2.
Okay.
Yeah, it's always going to be an NH2. Okay. There they are. And if you look at the next slide, there they are again. Okay, there's a whole big list. And this is hard to see, but you guys have it in front of you, and your eyes are young. I can't even read that list that you have in front of you. So you'll have to have these memorized by the test. I'm teasing. I don't know. Look, and that was worth the whole semester. No, you do not. I don't have any of them memorized at all. I don't care to memorize them. You do not have to. So, you guys will be getting a sheet attached to your test that is the carbohydrates, the monosaccharides, and you'll be getting this sheet right here, okay? It'll look just like this. It's not, without quite as much detail, I'm going to give you a sheet. It's going to have their structure and their name right here, okay? And I think on the sheet even, yes it is, you get the structure, you get the name, and the little 3-letter abbreviation for the name. Okay, I'll give you that, so don't bother memorizing these at all. All right, let's talk about the side chains. Let's go through our second list right here. You can see on the second list, and let's expand it up a little bit. There's some interesting things going on here. It says that a side chain, I told you the side chains will have another amine in them. And this list right here, the first thing it says is a side chain containing a non-basic nitrogen. So what we're looking at right here, let's get the pen, right there, side chains with nitrogen, but those nitrogens are non-basic. And if you look at them, can you guys see them? You see them okay? Okay? A basic nitrogen has a lone pair that is available to go out and grab things, right? That's the definition of nitrogen acting as a base. What do you notice about these particular amines? Like that one right there, for example, the top one that's the amide. What do you notice about the nitrogen's lone pair? It's involved in resonance, right? So because of that, it is not available. So it has it, sure, but it's not going to be basic. Next one, same deal. Not available. Now this next one, tryptophan, doesn't look too bad. It's got a lone pair sitting there. But it's under this non-basic list. So somehow that lone pair is not available. How come? The resonance, but more important, aromaticity. This lone pair is tied up in aromaticity. So that is absolutely not going to let go of its lone pair. Yes, this one right here, that NH2, that is part of the alpha amino acid. So that is basic somewhat. But we're talking here just when they classify the side chains is what that is. So these three side chains, even though they have amines, are counted as non-basic. Okay, they aren't going to be bases. Now let's go ahead and let's look at a side chain that's acidic. So we have, let's circle it here, acidic side chains, and then expand up. You can see all of them. All the side chains have COOH, COOH. All of them. And now let's go to a situation, the side chain actually is basic. And what we're going to be looking at with that, let's expand it up. You can see an NH2 there, and it's not, the lone pair is doing nothing. It's not trapped doing anything. Now this one's a little interesting. This unit right here is going to be your basic part. Well, I guess you could even expand it over into there, right? That's a weird looking thing, isn't it? Two nitrogens and then a nitrogen that is an imine. Which one would you guess is the basic nitrogen in there of the three? And think resonance on this. Two certainly are. If you look at the lone pair, let's just use this guy as an example, the end one. It feeds in, right? This one feeds in, right? That pushes electrons out onto that one, right? So which nitrogen has the most electron density of those three? The one that's the imine, right? This one, right, and let's do it in red. That right there has the largest delta minus. So that's going to be your basic one. And let's look at histidine. See if I can pull this up. Will it let me? Yeah, there we go. If we take a look at histidine right there. Oh boy. That to me looks like one of those nitrogens is trapped in aromaticity. Doesn't it look like that to you? Okay, so there's a lone pair and there's a lone pair. And we'll call this nitrogen 1 and nitrogen 2 for easy talking. Okay, so nitrogen 2, is that lone pair being used?
Yes.
Yeah, that lone pair is part of the aromatic system. So nitrogen 2 is not the one. Nitrogen 1, that lone pair there, it's not in the p orbital, part of that system, because the nitrogen already has a p orbital being used to form a pi bond. So that lone pair has to be in the sp2 orbital, which means it's available, it's not part of the aromatic system, that's the basic nitrogen. Okay, so far so good? Silence, I think, means yes. Okay. So, what we basically, in terms of the summary, 15 of the 20 are neutral. Even though they may have nitrogens in them, they're neutral. 2 have carboxylic acids, they're acidic. 3 of them that we just looked at have amino groups, and they are going to be basic side chains. All right, some of them are essential. You guys heard of the essential amino acids? Those are ones that your body is not able to make from other things. You cannot synthesize these. So they have to come from food sources. And there are, how many there? 10? 10 of them are essential amino acids. This apparently is, from what I've read, is something that vegetarians have to pay attention to. The meat eaters like myself, I get plenty of protein from all kinds of sources. And so I get all these things typically that I can ingest and break down. But vegetarians, some of these, I guess, are most commonly found in animal products. So they have to be very careful to ingest them from other sources or it causes trouble. Any vegetarians agree with that? You guys know? Yeah, you have to pay attention to that, right? Yeah. I only pay attention to how rare it is. That's all I do. Okay. Now, this is the no, not at all. Nope. Okay. Now, our last little gasp of chirality. All of the amino acids except glycine are chiral. So we are going to have one more torture session with R and S in this, which is good for you. So if it's chiral, that means a carbon has 4 different groups on it. And you can see with the amino acid that I showed right here that the carbon in the middle, that's just the alpha carbon, it does have 4 different things on it, so it is chiral. This happens to be an L amino acid, the one with the NH2 on the left. So an L amino acid will have the NH2 on the left. A D amino acid has the NH2 on the right. So it does track with the kind of nomenclature we learned for sugars, which is nice. D and L amino acids are enantiomers. You could draw a mirror plane down there. Turns out the amino acids found in most proteins, most naturally occurring ones, are L amino acids. There are naturally occurring D amino acids, but they're pretty rare. I read somewhere like a bacterial cell wall has them, that kind of stuff. But most of them are L. Now, amino acids are kind of interesting. This is getting into some chemistry. The way we've been drawing them, it's a carboxylic acid and an amine. So it should kind of be a normal organic compound. But it turns out they have really super high melting points, like really, really high. They tend to be, even with really alkyl-like side chains, like side chains with big alkyl groups, benzenes, things like that, they tend to be water soluble. And we have had experience with things with benzene. They are not water soluble, right? But these are. They have really large dipole moments, so they're extraordinarily polar. Which, granted, this thing is polar, but it's not like superpolar. These are superpolar. And they tend to be less acidic than a normal carboxylic acid, and a little less basic than a normal amine. So something's weird with them. All that behavior is characteristic of a salt. They act like a salt, not like a normal organic compound. And if you think about why that might be, it's because you have in the same molecule 2 functional groups that can interact with each other. You have a carboxylic acid and you have a nitrogen base. And so the nitrogen base picks up a proton from the carboxylic acid. And what you end up with is this thing called a Zwitter ion. You can see the nitrogen is protonated and the carboxylic acid is deprotonated. Which isn't, I don't think, a stretch, understanding their behavior, that would happen at all. So it's dipolar, so it's extremely polar. It is a salt. That explains its behavior. And so amino acids actually exist as this thing called a Zwitter ion. I don't know what that means in German. Means what?
Hybrid.
Hybrid. Zwitter means hybrid. Really? A hybrid ion. That makes sense. Okay. So you speak German. All right. So it's a dipolar ion. We lose an H plus in the acid, gain one on the amine. All right. Now, there's going to be, you're going to hear referred to this thing called an isoelectric point. And we're going to abbreviate that. It's like a pH, but we're going to call it PI. So this is the pH at which the amino acid exists as the Zwitter ion. So the isoelectric point is the pH at which the carboxylic acid is deprotonated and that nitrogen is protonated. So you have that dipolar ion. And where that point occurs, at what pH that occurs, depends on the structure of that side chain. If our side chain has a carboxylic acid, that's going to be a different effect than if it has an amine that's a base, which is different than if it's neutral. So let's think about this. If the amino acid has a neutral side chain, so it's like an isopropyl group, it's not doing much of anything, that isoelectric point occurs kind of around 7, but a little bit more acidic. It's about 5 to 6. So at pH 5 to 6, we'll have the Zwitter ion. If that amino acid is acidic, then, or the side chain is acidic, the isoelectric point is about 3, a little bit lower. And if it's basic, that isoelectric point is at about 9. It's slightly more basic. So acidic side chains, the isoelectric point's more to the acidic side. Basic side chain, the isoelectric point's more to the basic side. All right. So let's take a look at the next to understand this a little bit. All right. So what we've got here is a picture of an amino acid, and we are at essentially biological pH. And it has, if you take a look, it has a, let's see, our amino acid, here's our acidic side chain. So we have an acidic side chain right there. And I'm going to just go to the board and draw you guys a picture and give you some pKa values for these things that might help out a little bit understanding this. Okay. So there's carboxylic acid. And then let's do a CH, C double bond OH, and then NH3 plus. Okay. So looking at these amino acids, I went and asked our person who teaches our biochem about the pKa's of those carboxylic acids. And she said that the pKa of the alpha, the one that is on that alpha carbon, is at about 2 to 3. And so it goes first. It gets deprotonated first. The side chain one over here is at about 4 to 5. So it goes second. And then this one here is going to have to be basic. That's got a pKa probably about 9-ish or so. You have to be basic to pull that the hydrogen off the nitrogen. Okay, so if you take a look at what's going to go on as you raise the pH, so you start out acidic, everything is protonated. And let's go ahead and let's raise the pH. So I start to titrate with base. I'm going to pull off the most acidic hydrogen first. So when I raise the pH, I will have this structure. NH3 plus, and then I'll have that right there. And that is the Zwitter ion. That's what the Zwitter ion looks like. And so you can see from this picture here, if I keep raising it, we're going to get the one that's on the board. So add more base. Then I have a pK2, essentially. This would be pK1. From all protonated to this, I'm going to get the second one. And that amine will still be protonated. It's not basic enough yet for it. Then I get 2 carboxylic acids. This no longer is neutral. It's not a neutral ion. And so it's not the Zwitter ion. And then if I make it even more basic, add more base, this would be pK3 on this one. Then I've got it all. We get this. Then it's completely negative. So in this particular structure, it's neutral. When it was protonated, everything was positive. This structure, it's a net negative, it's a -1. And in this structure, it's a negative and it's -2. So for amino acids, you have at least 2 pKa's. Okay, at least 2. One would be the carboxylic acid. One would be that amine. And then, if your side chain is acidic or basic, you will have a third. You'll have a third pKa. All right. So, what we have here is biological pH where we're slightly above 7. That's basic enough that one carboxylic acid's gone and the other carboxylic acid is gone. But we're not basic enough yet for the amine. That one right there needs to be more basic to yank off that amine. So it's still protonated. If it's a basic amino acid, what we're going to see when we go on in a few slides is as I'm at biological pH, carboxylic acid's gone. At biological pH, it will always be gone. It's so acidic. But the amines, basic side chain, I have an amine there and an amine there. At pH 7, they are still protonated. So this thing would have a net positive charge. Okay. Next one. All right. So this slide is showing you an amino acid, and the side chain is neutral. We're just going to go with a neutral side chain. So initially, under acidic conditions, it has a positive charge, net positive. As we add one equivalent of base, just one equivalent, we yank off the carboxylic acid. This is the Zwitter ion. And then the second one, this thing has a net negative charge when we yank off the proton off the amine. Okay. Now let's go ahead and talk about how you could determine what these pKa values are, because remember they differ depending on what's on your side chain. Did you guys ever do an amino acid titration in like 401? You remember ever doing one? Yeah, just normal acid and base. Okay, it's a normal acid-base titration, but this is using an amino acid. You titrate it, so you add base, and you can figure out what your pKa values are by getting a titration curve, drawing a titration curve. I think they do this like in Chem 309. It's one of their exercises. So, in these curves, let's talk about interpreting this. You always start out fairly acidic. So this is the pH on the y-axis, and you are getting more basic as you go up. So up here is basic, down here is acidic. Along the x-axis, is equivalence of base added. So this particular amino acid, it's glycine. It's pretty boring. There's no side chain. So we're not going to have to worry about deprotonating a side chain at all. It's just the two main groups. So you start out these titrations really acidic. So we're down here at pH 0. So at pH 0, everything is protonated. So under acidic conditions, it looks like this. Below pH 2.3, everything's protonated. And then at pH 6, we have deprotonated that carboxylic acid. So that's our Zwitter ion. Now, as you're doing your titration curve, there is a point in between when you have added half an equivalent of base. At that point where I just drew the arrow, after half an equivalent of base, you have gone halfway. You deprotonated halfway. So what you have is you have a mixture of this CH2COOH. You've got that half, and you have the deprotonated version. The nitrogen's still protonated. It's a CH2COO minus. It's half this. If I've added half an equivalent of base, I've added enough that I've halfway deprotonated. That ends up, when you measure what that pH is on your pH meter, that ends up being a key point. You can see that they put a little star there and said that's pK1. You can tell what the pK1, which is the pK of this carboxylic acid, is by that halfway point. And the way you do that is with the equation. Remember Henderson-Hasselbalch?
Yeah.
Remember that equation? Yeah, what happened to that? Since you remember it, check me out. pH is pKa plus the log?
Yeah.
Yes? You don't remember it that well. What's the log of? It's base over acid, right? Not that it matters to us for this discussion. I think that's right. Base over acid?
Yes.
Yeah? Okay. So, at the halfway point, this becomes really important because here's the base form, right? Here's the acid form. At the halfway point, these right here are equal. And so they cancel one another out, right? If they're equal concentrations, they cancel. And the log of 1 is what?
0.
Right, 0. So at the halfway point, the pH that you measure on your meter is equal to that particular pKa. So I could give you guys a blank graph with nothing, no structures written on it, and it would just look like, say it looked something, I don't know, like, let's do this right, like this, and like this, and like that. And I could say, okay, tell me, and here's the amounts of base added, tell me what pK2 is. And pK2 would have to be halfway between this and the totally deprotonated, right? So pK2 would be right there at the 1.5 equivalent point. And you just measure its pH and you've got the value. So you can tell what the pK value is because it's always at the halfway points. So after a half a mole's added, that's our pK1. And we've got half of this and half of this. After one full equivalent is added, we have totally this completely, all this. So that pH is 6. After we add another half equivalent, we've deprotonated half of those guys. So that again is where we have half of this and half of the totally deprotonated. That's pK2, it's right there, about 9.6. Okay, and if there wasn't a side chain with a group on it, then there would be a third flat point, and we could tell what the acidity of that side chain was. Yes?
So that graph is really just has to.
This one would have, well, it should technically, I guess, yeah, the way I drew it, it did, because it would then go up like that. So that would be your third one right there. You can see that one just kind of disappears up to 14. Okay, so since these amino acids at different pHs have different forms, you can use that to separate them based on essentially their charge. And that process is called electrophoresis. So with electrophoresis, what you do is you dissolve the amino acid we'll just call it AA amino acid in a buffer solution. So it has a set pH. And you take a look at the form of the amino acid at that pH. And then you'll see that you could separate them. So let's say I had a mixture of alanine, aspartame, and lysine. And I have got the pictures of alanine, aspartame, and lysine right there. And let's go ahead and expand them up just a little bit. There they are. And we're doing this, by the way, at pH 6. So, just based on what we know, or just learned about side chains and what we're looking at, alanine at pH 6. Alanine has a neutral side chain. So pH 6 is about where the Zwitter-ion would be. So alanine, if you take a look at it, at pH 6, it is going to look just like this. And its overall charge is neutral. It is neutral. Has no charge. There's a plus on the nitrogen, a minus on the carboxylic acid. Aspartic acid rather, has 2 carboxylic acids. Now at pH 6, that's approaching basic. We're not quite there. But the pKa values of those 2 carboxylic acids, one was what, 3 and one was 5. So we're above that. So what that means is the carboxylic acids are both deprotonated, there and there. We've got them both. That pH is high enough, they both went. But it is not so high that the amine went. It's still protonated. So this thing, it's above its Zwitter-ion for sure, because it has a charge. It's not neutral. The overall charge is a minus, and a plus. It has a negative charge. Looking at lysine, there is a basic side chain. So at pH 6, I've gotten the carboxylic acid, it's gone. The amines, they have a pKa around 9, right? There's no way they're gone yet at pH 6. So they're still protonated. So I have a minus, and both amines protonated. Overall, that's a net positive charge. So lysine's positive. So if I dissolve this mixture of the 3 in a pH 6 buffer, one of them is neutral, one has a negative charge, one has a positive charge. So their ions are all different. Okay? Now, what you do is you take this thing, and we can fill in some of your blanks here, I think. Well, what is the form of each? You guys can fill it in. We just did down below. You take this thing, and you do an electrophoresis, which some people do, like in a plastic tub with electrodes at each end. Okay? So you take somehow and you get a gel, you smear in a stripe the mixture in the buffer on the gel, and then you have a cathode and an anode that you hook to a battery source. And you apply an electric current. And since the ions, some of them have charges, some will be attracted to the anode, some will be attracted to the cathode. Neutral ones aren't attracted to either, right? So let's go ahead and let's take a look at this. We're going to, in our first setup right here, we are going to be taking and applying a streak or a smear at pH 6. We're going to smear that mixture onto the gel. Then we're going to go ahead and we are going to apply a current. The anode is the one that has the positive. The cathode is the one that has the negative. Species that have a negative charge are going to be attracted to the positively charged electrode, because opposite charges attract. So the aspartic acid had the negative charge, and they're showing that here as ASP minus. So the aspartic acid, with its negative charge, moved out of its little streak and moved toward the positive electrode. Okay, the positive amino acid, which was the lysine, it had the positive charge, it was not going to move toward the positive electrode. It was going to move towards the negative one. So it moved out of its streak in the middle towards the negative. Alanine, which was neutral, stayed put, wasn't attracted to either one. So what you've done with this is you have caused them to separate on the basis of their charge and which electrode they're attracted to. Then you can separate, get them off the gel, wash them off, and then it's actually a separation method. Or you could use it as just a simple identification method too. Okay, you had a question, Ariel.
Oh, no, we just did this in bio.
Oh, did you? Did it work well? I've never actually done it. I've seen it like as a demo, but that was it.
Yeah, it was really cool.
Did you guys get them off the gel?
Yeah.
Okay, you did. All right.
Yeah, well, we were using bacteria. We were using bacteria, and then we had it run for about a day.
Oh, wow.
And
Okay.
then, because sometimes they come out smeary and
Yeah.
like streaks if you put too much in.
Yeah. Almost like a column. Comes out smeary if you put too much. Yeah.
Kind of like a meteor.
Yeah.
shower.
Okay. Oh, wow.
And so some people had really good ones, and other people had really bad
Really
ones.
bad. Yeah.
So,
It's cool how it works.
yeah, it was.
If they're both charged, like say they're both two of them are negative, the one that's more negative moves further. So you could get them all moving to the same side, just different graduations of movement as well. That can happen too. All right. Moving on. We'll just zip through this, then we'll call it quits at this point. Finish up on Monday, we'll finish up one way or another. Okay, as far as the synthesis of these amino acids go, they are actually syntheses that you already know. The first one is a synthesis from an alpha halo carboxylic acid. So you start out with a carboxylic acid, right there. You do Hell-Volhart-Zelinsky. So this first reaction right here is the HVZ. We just learned that a couple weeks ago. That's for alpha halogenation of a carboxylic acid, and it goes through an acid chloride. Remember that reaction? So you're really alpha halogenating an acid chloride. And you end up putting a bromine in the alpha position. And then it's just simply replacing that with NH3. It's an SN2 reaction. And the bromine gets kicked off, and you have an amine in the alpha position. Okay, so that's the first reaction. The one is a malonic ester synthesis, which we also learned. Okay, this malonic ester is a little bit different. You would never have to make this. You actually buy it. And the malonic ester has something attached at the alpha position. Does that look at all familiar, that group I'm circling? It's a protected amine. This was used in the Gabriel synthesis. Remember phthalimide? So this malonic ester has phthalimide stuck on it. I think it has an I right there. There we go, phthalimide. That just looks weird, but there it is. So it comes with a phthalimide on it. So it comes with its nitrogen attached. And what we're going to do at the very end is take the protecting group off, and voila, there's an NH2. Okay, that's the trick. So what we're trying to do is at the alpha carbon, we have the nitrogen, we need the R group. It's a simple malonic ester synthesis. We're going to treat it with base first. Then this group right here is whatever we want to put the R group on there. So you could do acid chloride, you could do here, they did an alkyl kind of halide with some stuff on it. You're going to put your R group on. So we have now attached to malonic ester the R group, and then H3O plus in heat is a deprotection step. We're going to deprotect the amine, and there it is. We get the NH2. So it's just a little variation of your malonic ester. And the last one, it's a little bit weird. It's the Stryker synthesis. And it's bizarre. You take an aldehyde, and you take ammonia, and you take HCN, so don't take any deep breaths, and you mix all that together. And what goes on is out pops at the other end your amino acid. So that aldehyde unit turns into, it adds a carboxylic acid and it adds an NH3. So here we go, stepwise. They gave the mechanism kindly for a generic R group. So this R group right there will be whatever your chain will be. That's going to be your side chain of the amino acid. So you, of course, would have to pick it appropriately. So when you treat it with the mixture, the NH3 and the HCN, the first thing you get is reaction with the NH3 on the carbonyl, and that's going to form an imine. And we've seen that before. An aldehyde plus an amine forms an imine. And then what happens is that imine does a reaction in this second step that is somewhat like, reminds me of cyanohydrin. Remember that mechanism? It's sort of a cyanohydrin kind of mechanism with an imine instead. So all we're doing here is we're protonating it. That makes it a better electrophile. And then CN- attacks, and you have an alpha amino nitrile. So I put a CN there. Very kind of cyanohydrin-like. But now if you look at the product, though, let's squeeze back down. Taking a look at that product, it's not a cyanide group. It is a carboxylic acid. Well, how did that happen? H3O plus, yep. You have to do a hydrolysis. So H3O plus in heat, and you've got yourself to your carboxylic acid. So it's all stuff you know, just slightly twisted. I will not make you memorize these. Okay? All I ask is that you can recognize them. I'm not going to say do the Stryker synthesis on the test. I won't do that to you. Okay? Just be able to recognize them. All right, so hopefully we'll finish up on Monday. The rest of it's pretty quick. If not, check D2L. It'll be there. We're going to go now into peptides. So we've studied amino acid, what are called monomers. That's just the little amino acid molecule. When you have a bunch of monomers pasted together, almost like a monosaccharide, two of them pasted together is a disaccharide, same kind of idea. We get a little bunch of monomers pasted together. We get this polymer, and they call those peptides. Now, to make a peptide, you take the amino group of one molecule, so that would be this group right here. So you're going to take the amino of one, and you are going to condense it with the acid group of another one. And you guys know enough about chemistry to know what that's going to do. You take an amine and a carboxylic acid. Synthetically, we used a lot of heat, right? And we talked about how this doesn't work very well because there's a lot of heat. Your body doesn't use a whole lot of heat. It uses enzymes and things to make it happen, catalysts to make it happen. We are going to do some what's called lasso chemistry, which they use in the lower level courses. We have never allowed you to do lasso chemistry. But what it is, I circled the OH, I circled the NH2, right? Lasso chemistry is where I'm going to put them together. Out flies, guess what? H2O, woo, it's gone. And we end up making an, let's see, there we go, an amide linkage. So we are creating an amide bond. So that's lasso chemistry. Now, I would expect that you could do a legitimate mechanism, right? This would not be acceptable, but it's not the purpose of what we're talking about. So water flies away magically, and we have an amide bond. In peptide chemistry and in proteins, they don't call them amide bonds. They call them peptide bonds. So a peptide bond is an amide bond. Carboxylic acid of one, amine of the other. Okay, each amino acid unit, so that's each little individual like monomer, is called a residue. So if I were to break the thing apart, say there were 5 amino acids all in a line, if I were to cleave each one, there would be 5 residues, and they would have their names associated with that. Yes?
So does that have to be this Witterson ions and
You know, it doesn't, but the amino acids exist as zwitterions. They're just showing that. They're just showing how it actually exists. We would, if we were making this synthetically on paper, we would not want that amine protonated because then it's not a nucleophile, right? So the enzyme magically does something to take care of that. Yep, for us, that wouldn't work. When you're talking about peptides, polypeptides, they have molecular weights less than 5,000. That's kind of small. But, you know, on our scale, that's pretty big. But peptides, generally, that's cut off about 5,000. When you get bigger, then you're talking about a protein. Proteins are huge. They'll have molecular weights 6,000 to, 40 million. So they're giant. So how come you do not weigh 40 million grams? You have proteins in you. You aren't going to need 40 million grams, though. You don't have a mole of them, right? Which is a heck of a lot of them. So that's what keeps us from being gigantic. Okay, questions on that slide? Yeah.
Also that whole molecule is one unit, right?
This whole, the thing, let me circle it here in red. This thing right here that I'm boxing there, that's one molecule now. It's a peptide molecule. Super teeny, but there it is. So far, so good? All right. Then let's go ahead and let's switch over. What I've got up there, it's a little hard to see because it's so light in here, but it is a nonapeptide. So just based on its name, you can tell that there are how many amino acid residues in there? 9, good. So there are 9 amino acid units in there. And you can see that there, if you look along, that we have amide linkages. There is an amide linkage right there. There is an amide linkage right there, an amide linkage there. And that's kind of the consistent thing with these is these amide linkages all along. They differ in what's hanging off of that alpha carbon. That's the side chain. So the side chains hanging off are what's making it different. It's a whole bunch of amide linkages. When you're drawing out a peptide, there's a convention to it. And the convention is that the end with the nitrogen, so here's the free nitrogen right here, is going to be on your left. You typically draw it on the left. That's called the N-terminus end. It's the N end, terminus or terminal end. So that's the nitrogen end on the left. The carboxylic acid, that is the other end of it, call it the C-terminus, that's typically drawn on the right. And you don't want to stray too much from this because these are named reading from left to right. So if you do it backwards, a person's going to get the order of the amino acids incorrect because they'll assume you're going N-terminus to C-terminus. So you don't want to be doing the opposite. Don't go against convention. Mike?
I only count 8.
You count only 8? Oh, let's see. We have this one at the end. Here's 1. There's 2. There's 3. There's 4. 5. 6. 7. 8. I count 9. Let's go. The N-terminus one counts too.
Oh, that's 9.
Yeah, so this one is 1, okay, and then 2 right here, 3 here, here's 4, 5, 6, 7, here's 8, and then 9. Okay, you see them in there? The N-terminus one counts. So this right here, if I were to like chop that off, that's 1 right there.
Okay, where's 2?
Okay? And 2 is right there. So it's a different color?
Different color.
Does that help?
Yeah.
Yeah, okay. That's a good, yeah, they did actually different colors, huh? So 1, 2, 3, 4, 5, 6, 7, 8, 9. So it goes, what, black, blue, like that? Okay? I never even noticed they did different colors. Are we good now? That there are 9? Okay. Now, let's just fill in some of these little gaps here. The N-terminus end is at the left, so fill in this one blank. Left-hand side, C-terminus is to the right. Peptides are named from left to right, and they use the little 3-letter abbreviation for them. So this particular one, I would write down ARG, that's the first one, and then a dash, and then pro, gly, phi, sir, pro's next, is that right? Phi, and then arg. And as long as you stay with convention, you could look up what these names represent. You could draw this peptide. Okay, just make the amide linkages where they belong. Yes? Yes. Yeah, the question was, would you need to do that for an exam? And yeah, I probably won't give you 9, but I may put up 3 and say name the peptide. And you would have to then take a look at this and say, okay, well this one is phi, this one is arg, that one's pro, so it's pro, phi, arg. So I would expect you, and I won't do 9, but 3, spare game 2 or 3, to be able to go from the structure to the name like this. Okay? No, it'll be on that piece of paper. You don't have to memorize them at all. You're going to get the amino acids, you'll get their structures, and their 3-letter designations on a piece of paper on the test.
Just like we're seeing?
Let's see. Just like you're seeing, you know that super tiny little graph that you could barely see? It was like that. Not as much detail, but I'll give you the picture of the amino acid, and I'll give you its 3-letter designation. So I'll tell you ARG looks like, you'll see ARG, and you'll see this picture right here. Just like that. Okay? So you don't have to memorize the amino acids at all. That'd be crazy. Someday you'll have to, but not for me. All right. Now, let's just pop to the next board for just a second. A big branch of biochemistry is peptide synthesis. My in-laws are actually both biochemists, and they work in research. And I went and toured, they were down at Texas A&M doing some biochemy thing. And I went and toured their lab. And both of them, their big deal was they would take mice, and they were trying to figure out some protein sequence that would cause like eye cancer or something in a mouse. I don't know. But what they would do is they would isolate what they thought was the protein doing it, sequence it, which we'll talk about in just a minute, and then they would want to build it. And I'm not quite sure, they'd take the chunk they thought was bad and splice it into things. So you need to be able to figure out, based on a given sequence, like what we just did with that one, you have to be able to build that, recreate it in a lab so that you could use it or test it or do whatever. So peptide synthesis is a huge branch of science. And what we're going to do is we'll pop over to the board, see if I can get this thing to just go away. I think I can. I may never get my picture back. Let's see. I want to get rid of it. It's never going to happen.
It's getting bigger.
It's getting bigger? That's totally not what I want. Well, let's see. I want this just to make it go. How about lamp? Go away. 2, no. Whatever. Here's how you make it go away.
There you go.
You turn it off. So what we're going to do is we're going to do a little peptide synthesis, and I'll show you guys how in a lab you would actually make this happen. It's kind of interesting. All right, so we're going to build a tiny peptide, a little 2 amino acid peptide. And we are going to make it out of phenylalanine. So we have H2N, CH, so double bond OH, and this is a CH2, and then a benzene. I'm just going to put the H on the benzene ring. Okay? And that is phenylalanine. And the other one's going to be gly. So H2N, CH2, C double bond OH. So this is gly. And what I want to build is I want to build the peptide phe glide. So I want to put these guys together, and I want to make phe glide. So in other words, phenylalanine will have the free NH2 at this end, right?
Right.
And glycine is going to have the carboxylic acid free. And that is exactly what I want. So imagine this, I go in, I throw it into a beaker, put, I don't know, acid catalyst or something in there, stir it all up, and I get phe glide. Okay? I also get glide phe because the molecule doesn't care that I want phe glide. Okay? It is
Yeah, we
unimportant.
noticed.
Yeah, that doesn't care what you want. It could also take, and it could take this carboxylic acid end and react it with that amine, right? All we need is a carboxylic acid and an amine. So we could get glide phe as well, which I don't want. We could also get what?
Like,
You get glide. And now you say it, what else? Ah, you guys are good. You could also get phe. And I don't want these things, right? So, think about this. How am I going to make sure that I do not get these? How am I going to make sure that only that and that react together? Only those ones.
Turn off one side of something.
Huh?
Turn off one side of
I gotta
something.
turn off the other ones, right? What was that? What's that called when I take the groups and hide them?
Protect.
I'm gonna protect them. So what you have to do is you have to protect the groups that should not react first. So I need to find a protecting group for an amine and a protecting group for a carboxylic acid. Protect them, then mix the groups together, and I will just have just the right groups to react, and that's it. So let's go ahead and let's do that, and we'll introduce you to the protecting groups as we do it. All right. For the nitrogen, we are going to be protecting the NH2 as a di-tert-butyl dicarbonate. Sounds scary. So we're going to use di-tert-butyl dicarbonate to protect it. And let's go ahead, and I'm just going to put up the reaction and then you'll see. So I have, let's just do RNH2. We don't need to draw the whole amino acid. There it is. There's the amine I want to protect. This di-tert-butyl dicarbonate looks like this. There it is. The bajillion oxygens in there. This has a name, because no one wants to write that over and over and over. It is called TBOC. So you treat it with TBOC. And we're going to be generating some acid. The amine needs to be deprotonated during this, so you put pyridine in there. When we had a bottle of this, and it was actually labeled BOC-ON, B-O-C-O-N, BOC-ON. BOC-ON, dude, right? So there it is. BOC-ON. And it puts a BOC group, which is that group, on the amine. So it's going to look like this. It looks amide-like. It's almost, if you want to think about it, putting it in terms of stuff we understand, it's like an anhydride. You see how it looks anhydride-like, sort of? It's very anhydride-like, so we're going to take the amine and get something that looks amide-like, but not quite, right? It's kind of like an amide. It's called a carbamate. So you get this thing, this BOC-protected amine. And then, let's just talk about its hydrolysis now. If I want to take it off, to deprotect, It's an H3O plus deprotection. So H3O plus will get me right back to my NH2. Okay, so that's an acid deprotection. So I can protect the amine of phenylalanine with Bach-on. Now I need to make sure the carboxylic acid of glide does not react. I need to block it. Carboxylic acids, they're pretty easy to protect. They make an ester. So you can either make, the two common ones are a methyl ester, so CH3OH, H+, be a methyl ester, or they will sometimes make a benzyl ester. So there's benzyl alcohol, H+. Whoops. There we go. There's the benzyl ester. The ester is going to be base catalyzed hydrolysis, and that will get us, let's have our little acidification step. That'll get us back to our carboxylic acid. So we can deprotect Bach with acid, and the conditions aren't strong enough. This will hydrolyze much more easily than the ester. So under the acidic conditions, we won't actually get the ester. The ester will hydrolyze with base, and base doesn't touch Bach. It's hot in there, isn't it? I'm not sure what this knob does. Let's find out. It's probably heat. Looks like we have no control. You guys just sit and cook. It's your last lecture, just be tough. Think of it as, it's like hot yoga, it's hot oakum. Okay. Yeah.
Can't you hydrolyze the ester using H3O?
Yeah, you can hydrolyze the ester with H3O plus, but you have to heat it. And it's harsher conditions. This hydrolyzes really easily.
Oh, okay.
So you can hydrolyze this very gently, it won't touch your ester. So, yeah, go ahead.
So we can use base and heat or acid, or do we have to do base and?
It depends what you want to do. So hang on to that question for just a minute, then I'll show you, okay? So what we're going to do with phenylalanine is we want the amine hidden. So we're going to Bach on. So I'm going to have Bach, NH, CH, CH2 phenyl, C double bond OH. It's protected. With gly, let's just make a methyl ester. I'll have the amine free. And I have OCH3. So now I've got the groups protected. And when I go and put these together, let's put a plus up here, there is only one possible thing that can happen, and that is reaction right here. Spitting out magically water. So I end up with a peptide that's protected at both ends. So there's the first one, CH2 phenyl, then my carbonyl, then my peptide bond, which is really an amide, and an ester. Now, my goal was not very ambitious. I wanted to create a 2-peptide long two amino acid long peptide. So I'm done. So what I would do here, I would treat it with acid, I would treat it with base, deprotect both, I'm done. Normally you want something longer than 2. You might want like 10, 20, 100. So if I wanted to add one more, let's say I wanted Phe-gly arg. Say I wanted that. I just built Phe and gly. How am I going to make Phe-gly arg?
Base, add.
Okay, for Phe-gly arg, I need to add arg right here. Right, everyone okay with that? So I need this guy free, right? I don't want this amine here free because it's gonna mess me up. It just parks right there. So what I would do, selectively deprotect one end. So I would treat it with base. So I have OH minus and heat. And then, we're not gonna redraw the whole thing, don't worry. This would give me Bach, and let's just do Bach, Phe, Gly, with a free carboxylic acid, okay? Everyone good with that? And then I would add to it Arg with the NH2 free and the methyl ester. So its carboxylic acid couldn't mess it up. Then I would build another one and another one. So you can just keep adding to whatever end you want, provided you deprotect the right end and the thing you added is protected correctly. And you can just keep going, going, going. Yes?
If the E is protected on the one side,
Uh-huh.
why will it still mess it up if the Arg carboxylic acid isn't?
No, I don't want the Arg. Let's see, the Arg carboxylic acid is, right? So it's out of the way, it can't react. So the only amine source I have is from the Arg.
But if we didn't protect the
Oh, if you didn't, it would mess it up because what I could get, it's not gonna react over here because this is protected. I could end up getting Arg, right? So I have to protect it. Huh?
What about phthalimide or whatever it's called? Is it a protection group of any?
Thalidomide it's not truly a protecting group. It's a way of putting a nitrogen onto something in sort of a protected fashion. It's more a synthetic tool than a protecting tool. So thalidomide itself is not used as a protecting group. It just occurs in things, bringing with it a nitrogen that it has to be unmasked. Yes?
Do we not do the H3O step if we're just deprotecting?
You would not want to do the H3O plus step. This right here will hydrolyze the ester, right?
Right.
But it won't do Bach. So if I were to do the H3O plus step, then I'm going to get this guy, and I don't want to.
So the H3O step isn't necessary.
The H3O step, you could, if you wanted to, I suppose you could just put this into, it could be into water, because there's water. Or I'm not sure, honestly, how harsh this has to be. Maybe if you got away with like 1 molar, maybe it hydrolyzes under 3 molar. Okay? All right, so you just build this thing sequentially, protecting, deprotecting, protecting the next group, then deprotecting as you need to. So imagine that you are doing one that's 100 long. That would be kind of a nightmare, right? Because you can't just keep adding acid, adding base, adding the next protected thing, adding acid, adding base. You're going to end up after like 4 steps with a soup of stuff, which is not going to work out. You would never run that. So what you have to do each time, if you're going to do it, you make this guy, you isolate it, you clean it up, then you do reaction 2. Then you would take the product of this, you would isolate it, you would clean it up, then you would do the next reaction, then you would isolate the 4 residue chunk, clean it up, and this could take you forever, right? Luckily though, they make this a little bit smarter. It's automated. No one actually sits there and does this by hand. So let's go ahead and let's take a look at the way that people actually do it that allows this process to be automated. And we need our screen back on. Essentially, we're going to be doing what I just described. Adding a chunk, cleaning it up. Adding a chunk, cleaning it up. But they made it a heck of a lot easier. Do we have light yet? Takes a second to warm up. Okay. Oh, it's counting, I see. Da-da-da. Voila! Okay. So this brings us to a solid phase peptide synthesis. What we're going to be doing with this is you have an attachment point, some polystyrene beads. So let's draw those like this. You have a line of beads, like a, I don't know, almost like a necklace if you want to think of it like that. And what you do is you're going to build from the carbon to the nitrogen terminus. So that chain is anchored onto the beads. You anchor the chain on, and then you anchor it here. You could have several of them anchored on there. So you have your growing chain anchored to these beads. And what you're going to do is you build it from the carbon to the nitrogen end, and you don't have to purify the intermediates in kind of the standard way. We don't have to like extract it and dry the reagent and evaporate, because what you can do is you just lift, come on now, you lift the beads out. The chain that you're growing is attached to it, right, dangling off of it. You just rinse it. You have a solvent rinse. That cleans it up. And then you go ahead, put it back into fresh solvent, and you keep building. So you can attach something, and then you just, I don't know, take a pair of tweezers, lift it up, hose it off, next beaker over, and there it goes. Okay? Still, people don't do this themselves. It's actually automated, and I think I have this is just a picture of the process. This is it's kind of a confusing picture. The solid support, let me come over here and point. The solid support is this purple box, right here. I don't know if it's purple in your guys', it's that box. You anchor the carbon in, the free carboxylic acid in. You start adding protected groups. These little triangles that are purple and yellow are protected groups. You start adding them, and you can just lift this out and hose all the rest of the stuff off of there, and then just continue on. So then you're adding your next protected group, and on it goes. Okay? So this is up on D2L in color too, if you want to see it in color.
So the protecting group is for the R side chain or what?
The protecting group's not for the side chain. It's either for the carboxylic acid or the amine end, depending on what you're trying to do. Okay? Now, as far as how it's done, there it is. A computer that you program in what you want, and a machine that synthesizes it. And I've actually seen these things in action. People program it in. Inside of it are a bunch of bottles. Down in here you see reagent bottles and things. Amino acids are down in there, acids are down in there, bases are down in there. You program in what it wants, and it makes noises, and it just kind of synthesizes it for you. It lifts the thing out, it rinses it, it puts it into the next solvent or solution that's needed, and you come back sometime later, and it's done. So it makes it a lot simpler. Yes?
Does the body do this by itself? Building this?
Oh, not quite like this. It's all enzymatic
But
in
it's
us.
Actually.
Yeah, I don't know if it does it in larger pieces, but it must, start out small.
So it's molecule by molecule that it's building? Really?
It's building molecule by molecule.
So it can only do.
But it's anchored, the chain, there are a bunch of them anchored on there.
Okay.
But yeah, it's doing it piece by piece by piece. Yeah,
Yeah.
pretty wild. Okay, so that's building it. This next part that we're going to talk about is how you would figure out what you want to build. I talked about how my in-laws isolate proteins and peptides from, rats, and they sequence what they think is the problem, the amino acid lineup that's the issue, and then they build it. So you have to be able to sequence it so that you can go and recreate the sequence, and that's peptide structure analysis. What you're going to be deducing is the order of the amino acids. That is what's called the primary structure. It's the most basic one. And that is just the order of the amino acids in a peptide. Now to do that, you need two pieces of data. You need the most obvious one, which amino acids are present, right? You have to know that. And you want to know how many of each type And then you have to find some way to figure out how they're bonded. That's the key thing, is you gotta know what they are and how they're bonded. So this kind of a curvy snake thing, it's a little bit hard to see, is a peptide chain. Each little circle is an amino acid. And you can see down here, they're showing a glow, kind of a blowout of the first four. And it's cis, sur, lu, and I can't read it, phi. Cis, sur, lu, and phi. Right there. And then obviously it would continue on. So if you want to try to replicate this, you have to know what the order is so that you can attach the right thing to your chain and have the right things added next. So let's chat about how to sequence and get structure. You're going to use something, and I thought this had to be a lie, an amino acid analyzer. That's like going to lab and say, yeah, we're going to use the ester maker. I mean, whatever. So it's called an amino acid analyzer. And what it does is it takes that long peptide and breaks it up and hydrolyzes it and tells you what is in there. So let's go through the process. The first thing it does is it takes the peptide and it breaks it into the amino acid residues. These are the individual pieces, the individual amino acids. So to do that, you're going to have to hydrolyze, obviously, the amide bonds. You're going to have to break those amide or peptide bonds. So then you've got floating around a whole bunch of individual amino acids. The next thing we're going to do is kind of sort them out. And we're going to do that by chromatography. So step 2, you separate the amino acids by chromatography. And you can see if we go, and I'm going to expand in on this picture here, see where we are in the picture. So we have up here a buffer solution, and this is where hydrolysis will take place. So this is where we're breaking them into the individual amino acid units. So all the little amino acids come traveling down this tube, go into a column. And as you have experienced with columns, they separate things, right, based on their ability to move through whatever gel is in there. So you have them moving through at different speeds, and they separate into bands. You get bands of different amino acids. Okay, it's like column chromatography in action. Yes?
So what if you have like a bunch of amino acids with neutral section?
They would travel through different than acidic ones and different than basic ones. And even the neutral ones will move slightly differently. Mm-hmm. What are you just going to ask me? No? Separate just into bands based on, just like in chromatography, like what we did. Oh, based on polarity, based on, it could be a mass, based on their ability to hydrogen bond with the resin that's in there. All sorts of the typical things. So then these bands come traveling out, the fractions, so you get fractions come out the end, and they mix the fractions with ninhydrin. I don't know if you guys, do you still use that in 401? It's a spray. No? No, it's kind of nasty. They might have quit using it. But you mix it with this ninhydrin solution. They don't use a spray. And what that causes is when an amino acid mixes with ninhydrin, so let's put plus an amino acid, you get a purple solution. So you get this purple solution, and then the purple solution, this is very complicated, flies into something that light shines through. It's almost like a spectrophotometer now. So light shines through the purple solution, and it will absorb light. So the solution will absorb light. So if there's no purple coming through, no light's absorbed, right? If a light purple solution comes through, then a little bit of light's absorbed. But let's say there's a lot of whatever that amino acid is in your peptide, a whole lot of it, there's going to be a pretty deep purple solution. It's going to absorb more light. Does that make sense? Okay, so you get to, it goes to a computer. And what the computer gives you, and let's move this guy over, is it gives you a graph. There is a time of elution. So this time is the time of elution. It's how long it took the thing to go through the column. Now remember your GC lab? You had standards, right? And you knew that chloroform took, I don't know, 1.2 minutes to come out, right? So if something took 1.2 minutes to come out, you knew it was chloroform, right? Remember that? Same thing here. They shoot through the 20 amino acids, they get the known times, they're essentially standards. Based on the time of evolution of your unknown, you can figure out what compound that was by matching it to the standards. Okay, just like the GC lab in that effect. So the time of evolution is directly related to identity. You just compare it to your standard knowns. Okay, so where something comes out, that's just like the GC lab. What do you think the height is related to? The amount, yeah. The height is related to the amount of the amino acid present. If it's taller, it absorbs more light. There's more of that amino acid. So this little graph gives you everything you need to know, just about. Tells you what's there, and tells you how much of it. Okay, now let's go look at a real one. I think that's our next slide. Kind of bring this guy back to life here. All right, one. Here's a real one. Along the top are the 20 amino acids run as a standard. So these are our standards right here. The bottom one is where an unknown essentially has been put through. And so we have 1, 2, 3, 5 peaks, so we know there are 5 different types. And just by looking at the standards, we can just line it up, and we know this peak, the first one, came out at the exact same time as the standard serine did. So it's got to be serine. Looking over here, this tall peak here, well, its time matched up with our standard phenylalanine. It's got to be phenylalanine. And I can also tell, just based on, the machine would give you how much area the peak has. I can tell there was more phe than there was sir in the molecule, because the peak's about twice as big or so. Okay? Yeah?
We can't figure out how they're attached like serve to the...
You cannot. At this point, I can't figure out how they're attached. I don't know that, because I just blew them apart to do this, right? So I don't know how they're attached. I do know what I have, and I do know how much of each one. Okay, but that's all I know right now. So I couldn't draw you a definitive peptide. I could draw you a bunch of possibilities, but nothing definitive. All right? So the next problem we have, and this is a little bit of a harder one, is figuring out this sequence. Okay, we've got to figure out in that chunk of 9 or 10 or whatever it was, how were they ordered? So that brings us to our next, to get the sequence. Yeah, Mike.
There are 20 essential
20
amino acids, but billions of others.
There are the 20 that are found in proteins and peptides are 20. 12, I think, of those are essential.
Oh, okay. So there's not an infinite number?
No, the alpha amino acids that are found biologically, there's 20 of them.
Oh, okay.
Okay? Okay, so to get the sequence, what we're going to do first is you're going to break that peptide chain into smaller, manageable fragments. So you're going to get smaller ones. So if it's 100 long, we might break it down in a controlled manner. We're just not going to like break it, but in a very controlled manner into chunks that are maybe like 20. Okay, much more usable chunks. Now, we want to do this in a controlled manner. So what that means is we are going to use enzymes to do it. There are catalogs of enzymes for doing this, very much like we have chemical catalogs. My in-laws had catalogs of enzymes. And they would look in there, and they would say, well, I want to cleave between lysine and arginine. I want to break just that linkage, that's all. So you go in there, and you buy trypsin. So you treat it with trypsin. You get pieces. You know those pieces cleaved at lysine and arginine only. So already you know something about the pieces, right? So you get these chunks in a controlled manner. And then what you do is you sequence each fragment so that your individual little chunklets, you're going to sequence those. And then you have a jigsaw puzzle. You know, okay, I had 5 pieces. I cleaved them between lysine and arginine. And in each of these 5 pieces, here are what was in those. And you'd sit there and play with it and try to find matches and try to find the order. Or what you really do, you feed it into a computer and walk away, and it sits there and crunches bajillions of possibilities and gives you the sequence. Okay, we're going to do a small one ourselves. And you'll kind of get an idea of why you want a computer when it gets bigger. Okay, now as far as sequencing goes, we have broken so far the 100 amino acid peptide into chunks of, let's say, 10 in a controlled fashion. So I have little 10 amino acid chunks now. To figure out the chunk, so I'm going to have, we'll use this one. It's a 10 amino acid chunk, so 3, 4, 5, 6, 7, 8, 9, 10. We're going to use the Edman degradation. And what the Edman degradation does is it cleaves off from the N-terminus end. So I cleave off the N-terminus amino acid, I get its identity, I figure out what it is. Then I do it again. So I know this guy now. I cleave this guy off.
Is that N-terminus?
I know, that's the N-terminus end.
Number 2 is.
Well, once I get rid of number 1, it sure is.
Oh.
Right? Number 1's gone. I have that. I cleave off number 2. I figure out what it is. Then I cleave off number 3. I figure out what it is. I cleave off number 4. I figure out what it is. It just keeps going chunk, 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 chunk from the N-terminus end. And each time it rips one off, you figure out what that one was. That's the Edman degradation. And let's go ahead and take a look at this. Action. It's a lot of structures. You don't have to memorize this. Okay, it's just going to show you how it works. So I have a peptide. This is a peptide. They actually showed for the reaction the N-terminal amino acid. Okay? So that's our orientation. We're going to treat it with phenyl isothiocyanate. That is what that thing is called. It's a benzene ring with an N double bonded to a C, double bonded to an S. It's phenyl isothiocyanate. I won't test you on that either. Then this weird thing happens. You could give me the mechanism if you were pressed, I'm sure. You get this 5-membered ring where you incorporate the amino acid and the phenyl isothiocyanate in there. When you hydrolyze it with H3O+, you break that 5-membered ring off. So now take a look. This is now separated from the molecule. It's gone. You analyze it with chromatography, compare it to standards, so you know what it is. So at this point, I know that I had, cystic acid at the end. This now becomes my unknown, so I've dug one in. Then I do it again. I take my new one, I treat it with phenyl isothiocyanate, hydrolyze it, I get this weird 5-membered ring, do chromatography, figure out what it was. Now I know what the second one in was. Then I do it again. Do the third, do the fourth, do the fifth. And I get an order that way. Okay? Very labor intensive. Yeah?
So the phenyl
Uh-huh.
isothiocyanate will cleave whatever the N-terminus is.
Exactly. It rips that N-terminus amino acid off. Just the one, though. And then you analyze it with chromatography. So you get an order. This particular example, we had a known peptide, it's unknown, analyzed the groups that were in it, got some chunks, and analyzed the chunks. So those are all the chunks that we obtained. And your job now, from this little nonapeptide, is to figure out what it was originally. So you start playing around with it. And the way I do this is typically, and let's just, I'll try to do it maybe on the iPad. We'll see how that goes. I pick one of them, and then I start seeing what I can fit around it. So let's start with, I don't know, the first one. So we will go ILEGLNASNCYS. Now, do you see that repeating anywhere else?
Yeah,
I see maybe the second one.
yes, or possibly. to go with it.
Yeah, the second one, look at what I see.
Too.
Yeah, I see ILE, GLN, and ASN. I'm seeing this part right here. You guys see that? So what came before ILE?
CYR.
TYR, right? And what else? Cis, CYS. So this is the second chunk right here, right there. See how there was some overlap? Okay. Now how about, does the next one help us at all?
The last one does.
GLN, ASN, CYS. I see this part right here. What's next? Pro. Okay. Prolugli. Yeah, probably, I'm guessing it'd be off of here. I'm kind of nervous about that though, because I only have one amino acid as my clue. So let's go down to the next one. Cis-prolugli. Am I more comfortable now? I'm seeing that. Now I'm happy. So pro, and let's go down here. Cis-pro, this has to be lu-gli. So there is my sequence right there. Now imagine there was 100 of these. Yeah. So what you do is you feed the chunks into a computer, and it does what we just did. Tries all the possibilities, then starts over and tries them all and starts over, and it finally gets a sequence. It's actually fun. This is my most favorite part of this entire chapter, is doing this. The ACS always has one of these, always does, on it. And it is 5 amino acids long. Okay, so don't let it tax your brain too much. It's only 5. Okay, everyone okay with that? All right, then let's go ahead to our thing. Now, proteins. Proteins are bigger. They are like hypercharged peptides. They have a lot of amino acid residues. There are different types of proteins. You can have simple ones, and those are just amino acids. That's it. That's all that's in there is amino acid residues joined up with peptide bonds. You can have ones that contain other compounds. Sugars might be interspersed in there, bonded to it. Those are conjugated, which is a very unfortunate word. You talk to an organic chemist, conjugated means something entirely different than something with a sugar molecule stuck to it. Okay? But they call those conjugated. You can have fibrous proteins, which are long, stringy filaments, like muscle and cartilage. That's fibrous. And then you could have ones that are more of a spherical, globular sort of shape. Those are called globular. Globular proteins are found in enzymes, hormones, things like that, metabolic processes. So now, let's talk about protein structure. Proteins are a little bit more complicated than just peptide bonds. So, what we're going to do is we are going to attack the 4 possible levels of structure that you're going to run into. The first one is simply the sequence in the long chain. That is the primary structure. So let's say you have 1,000 amino acid residues. The primary structure is just their sequence. How are they lined up? But then you get into shape. That long chain isn't just going to lay there 1,000, 2,000, 10,000 amino acids long and just go funk and lay there flat like that, like a snake. What can happen is it can curve. The groups over here can hydrogen bond to groups over here. That creates curves in the molecule. It can twist, held together with hydrogen bonds and things. So you get along this chain, not just laying like this, but kind of doing this and hydrogen bonding here and maybe hydrogen bonding there. You know, you get the idea. So it gets some shape to it. That is called secondary structure. So secondary structure looks at portions of that chain and sees how, like this section here, how's it twisted? What's it doing? That's secondary structure. And we're going to see 2 examples. It can twist into a helix in just a portion of the chain, or it could also make what's called a pleated sheet or a beta pleated sheet. And we'll see pictures of that. So I have this chain. It has an order. within the chain in little portions, it's kind of twisted and interacting with itself. But then it could be that this end where I'm standing twists way over and interacts with the end down there where Andrew is. So it starts to get these giant loops in it. And within those giant loops are little twists and things. So you have a big interaction, kind of a global interaction, with little interactions in it. Okay, so I might have a twisted chain looping over and touching way over there. That giant global sort of shape is tertiary structure. And we have pictures of all this, don't worry. And then, if I have one big chain, I'll twist it around and everything, and it interacts with other big chains, separate other big chains. That's called quaternary structure. So we're going to take a look at all of that in the next couple slides. We already did. Let's do secondary. With the first example of what a secondary structure can do, it could twist itself like a foam cord would, and that's called an alpha helix. So it's twisting essentially like this. And that would be in a section of the long chain. It would twist like that. The twists are held together with hydrogen bonds. So I would have, within here, some hydrogen bonding going on. So I might have an OH in one of my side chains, and the OH sees 4 molecules away a carbonyl and says, I'm just going to kind of hydrogen bond here. That's going to create a little kink in my chain fairly close up, and that's the alpha helix. It's like that. They also could interact and form a beta pleated sheet. This is a little bit tougher to see, these pleated sheets. What this picture isn't showing, it looks like individual chains interacting, but let's kind of draw some more to this. So here's your chain coming in, and you hit this one section that you can see here along the top. Okay, we hit this one section, and then this loops around. So your chain's still continuing. Okay, I haven't broken my chain, so it looped around, then it went through this section, and then it looped around here, and it went through this section and loops around. So it's my chain essentially forming layers like this, layering like that. Okay, but it's not breaking, it's not like that at all. Everyone all right with that? And it's different, it's not that twist, it's not like a foam cord, it's more like layers, like that, folded layers. Okay, so that's all occurring at small portions along the chain. Now the next level of structure is the chain acting further away. And that leads to tertiary structure. That's how the actual whole chain twists and curves with other parts pretty far away. You can see in this particular structure, this is a really long chain. Within it, I have a whole bunch of alpha helixes right there. So those are like little kind of local interactions. And then the chain globally is twisting around, and it's held through hydrogen bonds and things like that. It just coils all up. So what we're going to do now is I found on YouTube a pretty good little video that hopefully starts and plays for us nicely. Come on, go. It played in my office. Maybe we have to stop the recording.